So in this video, we're going to talk about neutrophils, which are a very abundant phagocyte that is used to phagocytose pathogens. So we're going to talk about how neutrophils work, where they're found, how they get to infected sites, what they do when they get there. So what should we remember about neutrophils? So usually when people talk about neutrophils, we're talking about this in the context of innate immunity. Recall, this is immunity that can act very quickly, minutes to hours to a day or two, when the pathogens are detected. And neutrophils are phagocytes. So they're part of the innate immune response. And like macrophages, they will phagocytose pathogens. Now, the difference, though, right, is that neutrophils are located in the bloodstream, right? And they're actually the most abundant leukocyte found in the bloodstream. Never let monkeys eat bananas. Uh, 50 billion found in the circulatory system at any given time, with even some more found in reserve in bone marrow. Whereas macrophages, which are also phagocytes, those are found resident in your tissues, and those are um, playing a slightly different role when they're phagocytosing pathogens, right? So neutrophils, they can be recruited into tissues, and that's typically where you find pathogens in our tissues. So neutrophils will move from the circulatory system into infected or inflamed tissues, and we're going to see that shortly. So neutrophils, they might be found currently in the bloodstream, but upon infection will migrate into the tissues. So again, neutrophils and macrophages are phagocytes, so they both have phagocytic receptors that will bind something on the surface of a pathogen and take that into the cell. Now, neutrophils are granulocytes, so they actually have inside them these clusters of compounds called granules, and that these can be used to neutralize the pathogen, and we'll see that in a bit. Neutrophils are short-lived. They will swarm to an infected tissue. They will phagocytose, use up their granules, and then die. And you actually have the formation of pus when you have a lot of neutrophil death going on. Whereas macrophages, they live in your tissues. They have actually lived very long lives, and when they process pathogens, they're actually taking those pathogens apart and using the fragments of the pathogens for other purposes, like MHC antigen presentation to naive T cells or other types of T cells. And we'll talk about that when we talk about cell-mediated immunity in later videos. But for now, let's talk about neutrophils and how they're going to migrate from the circulatory system into infected tissues, how they know where the pathogen is. So here is the bloodstream. There are neutrophils in there, very abundant, and there's no infection, so the neutrophils just keep swimming. Now, let's say there is an infection. So in the tissue, I've drawn some pathogens. How does the immune system know there's an infection? How does it recognize it? Often, this is through toll-like receptors, which we will cover in depth in a later video, that can bind molecules found on the surface of pathogens or released by pathogens. So macrophages can recognize an infection, and toll-like receptors will stimulate macrophages to produce cytokines, which are communication molecules, signaling molecules. We've talked about previously about TNF alpha or IL-1 beta, interleukin beta. These are two key inflammatory cytokines, pro-inflammatory cytokines. So they're going to help get this neutrophil into this infected site. And let's see how. So these two cytokines, we know, play an effect on the blood vessels, specifically the endothelial cells that make up the blood vessels. So on the surface of blood vessels, we have, on these endothelial cells, we have receptors for TNF alpha and I1 beta. And when they bind, they cause a change in the blood vessels, right? Remember from previous videos, we talked about vasodilation, increased vascular permeability. So bringing in more fluid, more proteins, more blood to the site. Well, something else that's going to happen to the endothelium, the lining of the blood vessels, is it's going to become activated. So what does that mean, activating your endothelium? Well, it's basically putting it in sort of an inflamed state so that the rest of the body knows, oh, there's an infection here, we should bring help. So what activated endothelium does, it can change its proteins on its surface. So now I have drawn I've drawn some proteins on the surface of these endothelial cells, and these are adhesion proteins. So ICAM1, ICAM2, these are two very common proteins that get turned on in response to inflammatory cytokines like TNF alpha and IL-1 beta. So now these proteins have ICAM1 and ICAM2 on their surface. So these are, again, proteins that are just, their genes have been turned on, the protein is made, the protein's expressed, sent to the surface of these cells, pointing toward the lumen of the blood vessel. And now we have these proteins sort of pointing out, and they're adhesion proteins. So something's going to stick to them. What is going to stick to them? There's a protein on the surface of neutrophils called LFA1. And these two proteins, ICAMs and LFA1, they interact. They interact quite tightly. So instead of neutrophils flowing by this inflamed tissue, these neutrophils actually stick to the walls of these blood vessels. Um, so this is what we refer to when we're talking about the activated endothelium. The neutrophils sense that there is an infection in the area. So they're going to enter this tissue. So that's what we talk about activated endothelium. So now these neutrophils will go into the tissue. How do they know to go into the tissue? They are attracted into there by another cytokine released by macrophages or other immune cells called CXCL8. If you recall, CXCL8 is a type of cytokine called a chemokine or chemoattractant. So cells such as neutrophils have a receptor on their surface for CXCL8. So that's the CXCL8 receptor. And what neutrophils will do, because this is a chemoattractant, like as we said before, is they actually will migrate toward high concentrations of the cytokine. So these neutrophils, they have stopped and bound to the endothelium, the activated endothelium, and now they're going to go squeeze between the endothelial cells. And again, remember, with increased vascular permeability, so these neutrophils can make their way through the line, through the walls of the blood vessels. We call this diapedesis. So we're moving neutrophils from the circulatory system into tissues, right? And so now neutrophils are in tissues. So we refer to this as extravasation. They have now crossed from the circulatory system into tissues. And now that they're in tissues, they're going to detect and phagocytose those pathogens. Before we get to that, let's also recall that uh, many pathogens can activate complement through a variety of pathways. We've got the alternative, the lectin, the classical pathway. Either way, in any of those pathways, we recall that complement protein C5 can get cleaved by C5 convertases into C5a and C5b. We know C5b helps initiate the formation of the membrane to tag complex. C5a is an anaphytoxin, helps get the inflammatory response and help recruit immune cells to the site of infection. So if complement is also being activated in this tissue, C5a is being formed. If C5a is being formed, that can also attract neutrophils. So neutrophils have a C5a receptor, and C5a can act also as a chemoattractant for neutrophils. So neutrophils will go and move toward the high concentration of C5a. C5a is going to do something else. We'll see on the next slide. So you can see here how neutrophils have now gone from the circulatory system into infected tissues. There we go. Okay, so now neutrophils are in the infected tissues. How do they know what to phagocytose? Right, they're phagocytes. So let's see. So neutrophils, like macrophages, have phagocytic receptors. So they share some phagocytic receptors. And so if you recall in the video on macrophages, we introduced the LPS receptor, which is also found on neutrophils. LPS receptor will bind LPS, lipopolysaccharide, found on the surface of some bacteria. And if that's the case, then LPS receptors will bring in this pathogen via receptor-mediated endocytosis, right? Also known as phagocytosis. There are mannose receptors on the surface of neutrophils. Mannose is a sugar found on the surface of some pathogens. And if mannose receptor binds mannose on the surface of the pathogens, help bring that pathogen inside the cell. So, and again, another phagocytic receptor. CR3 and CR4 are complement receptors. So if a pathogen is covered in complement, specifically IC3B, then this neutrophil can phagocytose this pathogen. And remember, this is an example of opsonization, when complement becomes affixed to the surface of a pathogen, and that makes the pathogen more attractive to neutrophils. So if this pathogen didn't have LPS on the surface, didn't have the mannose on the surface, but one of the pathways of complement activated on the surface of the pathogen and affixed C3Bs, complement fixation, that's going to trigger receptor-mediated endocytosis via opsonization. As we saw in the last video, we mentioned that neutrophils have a C5a receptor, right, which is C5a, it's an aflatoxin, helps provoke and stimulate the inflammatory and immune response. So one other thing that C5a will do when it binds neutrophils will send a signal into that neutrophil and actually get it to boost the amount of complement receptors, CR3 and CR4, which you would think may actually make sense, because if complement's getting cleaved and activated and we're forming C5as, then there's probably complement fixation going on the surface of pathogens. So we're going to phagocytose those pathogens. Great. How do neutrophils work to destroy pathogens? So neutrophils take in pathogens via receptor-mediated receptor-mediated endocytosis, which we call phagocytosis. So we got a phagosome vesicle in the neutrophil. Neutrophils are granulocytes, so there are these granules that contain a number of very toxic substances, and these granules will fuse with the phagosome and release these very toxic substances into the phagosome where the pathogen is found. So what kind of things are in there? Proteases, peptides that can disrupt plasma membranes, hydrogen peroxide, peroxidases, highly reactive oxygen species that will just interact with molecules within the vesicle, and that will cause those, the pathogen really to become non-functional, but basically interfere with its metabolic activity, interfere with its proteins and its genetic material, so that they won't be able to carry out functions. There are also within neutrophils lysosomes. When these are vesicles that are also contain toxins, enzymes. Then those get fused. The vesicle gets fused with the phagosome, and those toxins and enzymes get released, and again help destroy the pathogens. So we got a lot of killing going on in here. Once the neutrophil uses up its granules, it dies. It doesn't replenish the granules. So neutrophils either undergo apoptosis, cell death, so they'll destroy themselves and anything in them, or they can actually undergo a type of apoptosis called netosis, which is a very interesting mechanism whereby the neutrophil will create nets, almost like spider webs, to catch pathogens that might be in the area. So these nets literally stand for neutrophil extracellular traps. So during the process of while the cell is dying, it can actually take its genetic material, its DNA, and DNA is usually very compact, it's condensed. It will decondense the DNA and the nucleosomes and actually will release it outside the cell. So this is like a net, literal net. And so this decondensed DNA with proteins stuck to it, like proteases and nucleases and nucleosomes and histones, really acts like a trap to trap up and interact with pathogens. So it helps capture the pathogens. And again, you have all sorts of other immune cells in the area, like more neutrophils, like macrophages, doing their job to kill the pathogens. So these are just some mechanisms by which neutrophils will actually neutralize a pathogen.